So thank you all for staying with us until now. We are in the home stretch. This is the last session where, from an economist point of view, the rubber meets the road. We are going to talk about business models. And actually, maybe we can wait till the folks are seated in the back. And can we shut the doors just so that the folks who are talking outside can have privacy and we can as well? I've heard informally from a number of you during the course of the last few hours that one of the biggest challenges you see is the business model for rare diseases. And not only am I here to tell you that that is not nearly as much of a challenge as you think, but I actually have experts on the panel who can tell you that directly from their experience. So the way we're going to organize this is we will have a couple of comments to get us going about the backdrop of rare diseases, and then the kind of therapeutics that can be developed and the investment models. And then we're going to dive into various different business models beyond that. And so we're going to try to do this as quickly and as efficiently as possible to have as much time for Q&A, because I think that's really where a lot of the value is going to be gotten in this last session. It's answering your questions and addressing your issues in how we take some of these various different business models and apply them to appendix cancer. So with that said, let me start by asking Marshall to take the podium and give us a little bit of background about rare diseases.
And thank you. And see if you still want to do that in a few minutes. In the words of Monty Python, now for something completely different. Hang on, let's go back there. So first off, I'd like to welcome you to the field of rare disease. I've been doing it academically for about 40 years. I just joined the private sector recently. There are some interesting things about the field that y'all should probably know. Many of you have dealt with rare disease. Chris Austin dealt with it for years at NCATS, and we used to have a lot of these conversations, but there's some things you should remember. A lot of these, now, while many cancers may be somatic mutations, a lot of them are also germline. So these are lifetime diagnoses. These are things that patients have from birth going all the way through. There's limited incidence data. There's limited outcome data. You will have tremendous genetic heterogeneity across patients where you will have very early presentations or very late. Patients and families are often your best sources of information, and something that most folks in the regular medical world are very uncomfortable with, having a family that has more expertise around a disease than you do. They'll use multiple specialties, more than almost any other area of medicine, and care expertise is quite limited. And there's also kind of a little thought thing. In general medicine, many paths can lead to a diagnosis like hypertension, lots of causes. In genetic disease or genetic rare disease, usually you have a point of origin that can then lead to many clinical manifestations, but you have that common origin of a genetic defect. And that's actually a little bit different way of thinking of things. Defining what it is depends on where you are geographically. United States, we tend to stick with the FDA definition of under 200,000. The European Union uses 1 in 2,000. Taiwan and Japan uses a 1 in 10,000, but then they only have listed diseases. Western Australia, 1 in 2,000. But a good general guideline for rare disease is something a general pediatrician or a general practitioner would not be expected to know much about. They might have heard the word, but they really wouldn't know much about it. And I certainly think appendiceal cancer probably fits that. Growth in recognized diseases. Since I had fun, I got to be part of the Human Genome Project way back when I was younger, which I was at one point in time. The number of conditions now where we map a genetic defect to a clinical phenotype is over 10,000. Unofficially, it's probably around 12 to 13. NIH now says over 10,000. Even if we use the number where we've got those listed in our data sets, which is about 7,000, how many new conditions a week is that since we finished the genome? It's about 6 new diseases a week that we are describing. These are not new in the population. It's just that now we're describing their molecular defects. There's no other field of medicine like that. That means we are swimming in a vast sea of ignorance, and we have to be comfortable with that. It also means that we use informatics more than any other field of medicine. The other thing that's changing is prevalence. Let's take Down syndrome as a great example of that. 1980s, average life expectancy is about 25 years. It's approaching 60 now because of changes in treatment. Cystic fibrosis, sickle cell anemia. So not only are we recognizing more conditions, we're doing more treatment, patients are living longer. Some of those treatments are new drugs, some of them though are just getting organized and recognizing the conditions. What about in healthcare impact? So took the ICD-10 list of rare conditions and applied it to 1.5 million visits at a children's hospital, actually Children's National where I'm from. If you have one visit to the hospital, outpatient, inpatient, you've got about almost a 12-13% chance of having a rare disease diagnosis. If you get admitted, it's about 20%. If you make 2 visits to a children's hospital, even if it's to go into the ER and get an ear checked out, you got about a 30% chance of having a rare disease ICD-10 diagnosis. So these things have a huge impact on the finances in the pediatric world, and we're now starting to see that show up in the adult medicine world. I think the percentage of world drug market is about 18 to 20%, Andrew, for orphan drugs at this point in time. So while there's 10,000 conditions, each one fairly small, as an aggregate group, it's one of the most impactful areas of medicine. We have labor shortages. That's the other thing we run into. We have about 1 geneticist for every 250,000 in the US. Elsewhere in the world, it's about 1 to 3 to 4 million. So we don't have a lot of folks who have kind of grown up knowing how to do these things. So we have to expand into other areas to pull in expertise, but realizing that it's kind of an unusual field. So what sort of things are happening to deal with this? We have the National Organization for Rare Disorders, Clinical Centers of Excellence in Rare Disease. That's 32 sites. Boston Children's is one. We have the RareCap program, which is a project coming out of Vanderbilt University and Children's National on standardizing protocols. When you have 6 new diseases a week, you can't do your classic, let's wait till the literature matures and then all of our treatments will be done from the literature, because there's no literature. So you actually have to use a lot of common sense and a lot of physician experience. The I Am Rare program at NORD, which is a great, cost-effective natural history, and I think that's one that you all will be engaged with, which is great. It's a really good program. Then there's some great training programs as well, too. Costs for treating an orphan disease run about $97,000 a year, compared to most diseases around $15,000 per year. Lots of new therapies. They're all over the place with different things coming on. What's kind of fun for me now is a lot of the things we've been using in rare disease, you're starting to see in more mainstream medicine, starting to see use of some of the genetic technologies, things like that. FDA orphan drug approvals, I think probably one of the most successful incentive programs in history from 1983. So this is the 50th anniversary of the Orphan Drug Act, by the way, yay. Over the last 5 years, about 100 approvals every year for orphan drugs, so over 1,000 drugs approved. It's about 500 unique drugs. About 52% of the drugs approved by FDA over the last 3 or 4 years have been for orphan indications. Half of those are for cancer. So cancer, as always, is doing pretty well for itself from that standpoint. But even with that, a quarter of the drugs are for what we would call true classic rare disease. So there's a really good pathways to trod there. And there's a lot, the rules are kind of different from your classic FDA approval process. Early discussions, early interactions are definitely recommended. One reason that it's been so successful is the odds of success are higher. Odds of an orphan drug making it through approval are about 26%, which are lower in other areas. Cancers and oncology obviously have a somewhat much lower rate. And this is kind of the landscape right now for the drugs. So while orphan diseases or rare diseases are uncommon, the number of drugs coming through the process right now is not. All right, I'm going to close with this one. One of the things we've been working on is how do you improve the efficiency of the process. The classic rare disease clinical trial has about half the patients or less than a regular trial, but takes about twice the time. Takes on average 6 to 7 years longer than a standard disease. So while the denominator is quite low for these. The numerator is quite large. For these, average cost for an orphan drug to come through is still about 500 million. So we've been working on some plans and some things, a little thing called uncommon cures, to see if we can actually lower that and shorten the process. So we may get into that a little bit later. All right, let's go to the next.
Marshall, just a couple
Certainly.
of sentences about your background, please.
Oh, certainly. I am a career medical geneticist. I'm a biochemical and clinical geneticist and a pediatrician. Trained at Vanderbilt University many years ago in the mid-'80s. Spent most of my career there, developed the biochemical genetics program, and then also has played a lot in the BioView program there, for some of the large data sets and large genomic data sets there. Moved to Children's National in 2010, where I ran the Rare Disease Institute, which we built. And when I stepped down last year, it was the world's largest clinical genetics program.
Thank you. Steve?
All right, hi everyone. I'll start with background. Oh, there we go. There are the slides. Steve Curtis, Director of New Ventures at BrightEdge, which is the impact venture capital arm of the American Cancer Society. Andrew, thank you so much for the invitation to speak and the opportunity to contribute to this conversation today. I'll start just by a quick personal background. So I've been with ACS for a little over 7 months now, so relatively new to the team. The effort that is BrightEdge, though, is also a relatively new effort for American Cancer Society, so I can talk about that a little bit more. I've been in the venture capital, business development, big pharma, small biotech sort of space for most of my career since finishing my PhD in cancer genetics, other side of the river at Harvard Medical. Most recently was at MPM Capital for nearly 5 years, managing a seed fund collaboration we had with the Dana-Farber Cancer Institute, but also participated in some of the other funds that MPM also manages. To set the stage a little bit more specifically for oncology, just to expand on what Marshall has already kind of laid out here, just wanted to highlight just a couple slides from a study that has been done and overseen by Dr. Ahmadine Jamal, who leads our Cancer Surveillance and Health Equity Sciences division at American Cancer Society within our discovery pillar. A few different studies that are relevant to rare cancer specifically from 2014, a focus on childhood and adolescent cancers. In 2017, specifically a focus on rare cancers in adults. And then more recently from 2020, the cancer in adolescents and young adults. These are all subsections or sort of focus areas from our Cancer Facts and Figures that is put out every year and updates sort of the state of the nation on how we're doing on the fight against cancer. Couple definitions. NCI defines a rare cancer as less than 15 cases per 100 people in the US. The Rare Cancer Coalition has a slightly more stringent definition of that with less than 6 per 100,000. And that's actually the definition that ACS has adopted for cataloging this issue. Couple things that I think are incredibly striking from these data. There are nearly 200 different types of rare cancers that have been identified. I'm sure that number is already out of date, and you can add a few more to that already. Over 100 of these are very rare, so less than a half of a case per, or 1 case per 200,000. Interestingly, most of them show up in pediatric and adolescent cases, right? So that's where you're seeing the most prevalence of rare cancers. But it's this box in red that I think is the most striking. Like 1 in every 5 cancer diagnoses is considered rare. That is a big issue when you add it all up. So while, again, I think Marshall made the point very, very articulately, right? It's not that rare cancers or rare diseases are uncommon. They're actually quite common. And understanding new models to make them attractive business propositions to treat those patients that need it the most is a really important issue. So glad to be part of this panel and to participate in this conversation. Thank you.
Thank you.
Hi, I'm Jennifer Levin Carter. I am currently a venture partner at Sandbox Industries. We're the managing partner for the Blue Venture Fund, which is the venture fund for the Blue Cross Blue Shield plans. My background is I'm a physician by training. I started my career at Harvard Med School, practiced up here in internal medicine for a little while, And then I started one of the early precision medicine companies called N of One. We became the world leaders in what today is known as molecular oncology decision support. Sold that company to QIAGEN, started another kind of company in the clinical trial space, tried to reinvent the process of clinical trials, sold that company pre-launch to Flagship Pioneering, and ran Precision Health at one of their companies for a little while until they shifted more preclinical. And then, in addition to being involved in many precision medicine companies and a variety of other healthcare companies, I joined Sandbox Industries in the Blue Venture Fund. And part of the reason that I did that was because one of the observations I made during my journey at N of One and beyond was that one of the hardest things about getting these types of drugs to patients and even the diagnostics to patients. You can't even get the drugs to patients until you understand the biology of their disease. And to understand the biology of the disease, they have to get access to diagnostics, which is unfortunately the ugly stepchild in the whole process of clinical medicine, I think. But one of the biggest barriers is reimbursement. And so having been in the field for 15 years, I knew nothing about how the payers think about reimbursement. And we are starting a fund to really look at how we get payers and providers and industry to collaborate to drive the adoption of innovative technologies through venture capital. And so that's my background. It's an exciting opportunity, but quite frankly, I think that we're not even defining rare cancer the right way, because actually all cancers are rare. So if we just take non-small cell lung cancer as an example, back in 2008 when I started N of One, it was called non-small cell lung cancer. And now there are probably, I would say, at least 15, 20 subtypes of non-small cell lung cancer. And right now, there's a drug on the market that targets almost every one of those subtypes of lung cancer. It's really remarkable. Similarly for breast cancer. But I would argue that almost every cancer type is rare. And so our definitions of rare disease and rare cancer have to really evolve because we can understand them at different levels. And one of the things we saw today was, well, we're pretty used to using DNA based technologies to understand the biology, but actually even those aren't really paid for today for the majority of cancer types. And now we have RNA, and we've got proteomics, and we've got a lot of different ways, phosphoproteomics. I mean, so how are we going to bring this all together to benefit patients? And that's really something that I'm really interested in trying to solve. And some of the ways that we pay for drugs, that we pay for diagnostics, these are some of the really fundamental problems in our system. And I look forward to chatting about some of that today. Thanks.
Hello, I'm Fernando Vieira, and I have a confession. I don't know anything about cancer. I don't do cancer research at all. That's not true. I know some about cancer. Like everybody in the United States and in the world, somebody that I've known and loved has been affected by cancer, or I've lost people to cancer. So we all do know some. But my background, I work in ALS, amyotrophic lateral sclerosis. I'm an MD by training, but I don't practice. I've been doing drug discovery, drug development, and translational research for ALS for about 20 years. Now I serve as the CEO and the Chief Science Officer at the ALS Therapy Development Institute. If you don't know what ALS is, it's amyotrophic lateral sclerosis, also known as Lou Gehrig's disease, also known as that ice bucket thing from a while ago. It's a disease that affects both upper and lower motor neurons. A person who's affected by ALS loses their ability to move, speak, eat, swallow, and ultimately breathe. It's universally fatal, and people pass away within 2 to 5 years of their diagnosis. So it is a devastating condition. Like I said, I'm the CEO and CSO at ALS TDI. ALS TDI is a unique organization. It's a nonprofit biotech. It is it's on purpose nonprofit biotech. It's really a fantastic business model, in my opinion, particularly for a rare disease. ALS is a rare disease. It's probably a collection of ultra rare diseases, and there's a lot unknown about it, and there's a lot that we have to sort out. So it's probably going to take a while to knock it out completely, to really address all of the needs of all of the people affected by ALS. And so the nonprofit business model kind of marries two different approaches. You have the mission of a nonprofit, the focus that you're never going to stop working on that specific disease. It doesn't matter if you identify or invent an interesting technology that could be leveraged for another condition, fine, you give that to somebody else and they figure that out. We stay focused on ALS. We also have very, very deep roots in the ALS patient community. We interact every day with people who have the disease, and they're our driving force, and they also inform a lot of the research that we do. On the other hand, the nonprofit, sort of the biotech side of things, we're not a typical nonprofit in that we're very product-oriented. We're trying to invent and advance drugs. It's not basic research necessarily. It's more about how do we get something to people with ALS as quickly as possible. The way it works in some ways is that we're funded by philanthropy, grassroots fundraising, grants from foundations, some federal grants as well, peer-reviewed grants, and partnerships with for-profit entities. We do fee-for-service work in some cases, but mostly it's philanthropy and grassroots fundraising, and that gives us essentially a Series A every year, right? A small, non-dilutive Series A, and we get to use that to discover. But it also gives us the opportunity to build institutional knowledge over time, right? So we have team members who have been with us, who've been with me, gosh, poor people, for like 20 years, right, working together. And that goes a long way for diseases where there's so much unknown, like ALS. So what this has done, what we've been able to accomplish with this over the past, decade plus, is about $150 million raised over that time. We've been able to contribute to the advancement of 2 drugs that are now in phase 2 of ALS clinical development, one of them being developed for other indications as well by a for-profit partner. And we have 2 really promising programs in preclinical development right now that we're moving toward IND enabling. So it's a unique model. It's a brick and mortar lab in Watertown, Massachusetts. You're all welcome to come visit. We've got 15,000 square feet of lab and 30 scientists working full-time every day doing ALS drug discovery research. But yeah, I think there's opportunity for something like this to exist in the cancer space too, and happy to contribute to the conversation today. Thank you.
Yes, thank you. Tom Trimarchi, Chief Product Officer at BridgeBio Pharma. First, I want to thank Andrew and all of you for the opportunity to come here. It's a real privilege and honor to be here with you. Just briefly on my background, I started out as a scientist. I did a PhD in pathology at NYU. I then did my informal business training at a bank called Goldman Sachs for a few years as an equity analyst. And then I made a hard turn toward the corporate side, where I first jumped into Regeneron in the strategy group, and then quickly thereafter found out about a startup called BridgeBio, which just had a really compelling vision and really interesting story behind it. Maybe I'll kind of start there by telling you a bit about what we do and the business structure, and also the unique financial
forms of capital that we've been able to attract as a result. The story really starts with some a brilliant work out of Dr. Lowe's lab, which suggested that there is this valley of death that we all are aware of, where you've got tremendous innovation happening in academia, but very little of that is actually being translated into company-sponsored R&D pipeline programs. So why is that? I think as Andrew got it at the beginning of the day, you have issues with starting this early. So fundamentally, when you make a discovery in your lab, that is the earliest you can possibly start on a drug program. You just made a discovery. We don't know if it's going to replicate in another lab. We don't know if it's going to replicate in an industry lab. That's day one for a program. So the issues with any one of these from an industry perspective for an investment is it's the time when the probability of success is the lowest. It's the time when the time to market is the longest and the dollars required are the greatest. So when you put that on top of the concept that some of these diseases that we like to focus on are rare and the end markets might be limited or even unknown, that makes for a challenging investment case in isolation. But the brilliant idea of applying the financial principle of diversification to this problem tells you that if you put enough of these together, you reach a critical point where the risk of any one of those is removed and the investor can actually get a good return per unit of risk, and the odds of succeeding on any one of these is very high. To put it another way, at Bridge, we like to talk about it as this is a lottery ticket industry where the odds of failure on any project, as we heard from the keynote at lunch, is very high. So if you have a 1 in 10 chance of succeeding, you can't take one shot because you're not going to hit. You'd be very lucky for that to work. So you got to take a lot of shots. And so when you have a big portfolio, your odds of succeeding on one of these programs, which can make up for all the losses, is very high. That has the other interesting property of attracting non-traditional forms of capital into an R&D stage biotech company. So traditionally, you have an idea for a startup, you go raise venture capital equity, you go raise public equity through your IPO, and it's equity, 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 burn, burn, burn until you get acquired. Or if you're one of the rare cases that makes it, you actually become profitable. With our portfolio approach, we actually were able to attract several different forms of non-equity capital and equity capital. We since our inception, I think we raised about $1 billion of equity, including one of the bigger IPOs of the last few years. But we also were able to tap almost $2 billion in debt capital, which interestingly, the whole story of the diversified portfolio resonated much more with those types of investors than your typical equity investors. So that was kind of an interesting learning. So those are kind of the different business structures and financial kind of opportunities that we've enjoyed through it. And maybe just kind of a few words on where are we? Has it worked? How's the pipeline progress going? I think the whole question when we started was, would the theory of the case play out into actual practice? So I told you the issues with the valley of death are probability of success, cost, and timelines. Those are the negative things with any one program. So we wanted to think about how could we, aside from having the diversification benefit, how could we reduce all of those elements in our structure? Probability of success, we chose to focus exclusively on genetic diseases. So that's Mendelian and cancers with clear genetic drivers. And that means some rare cancers, some not so rare cancers. The other thing we do is we only go after diseases where we call them blueprint diseases. So it's where you can draw the line between the genetic defect, exactly quantitatively what it does to signaling, and so on until you get to patient symptomatology. We call them more like engineering problems. Second, we only go after diseases where we think we can hit the disease directly at its genetic source or immediately proximal to that. The reason for doing that is because we think that's going to make you have both a large effect size, high probability of success, and perhaps lower toxicity risk, although that's kind of speculative and intuitive. On the cost and timeline issues, we've designed our organization to reduce costs and accelerate timelines, so to gain efficiency, as we say. And the way we thought to do that was actually to employ a decentralized model where we don't have big R&D divisions, every project is a small, focused, dedicated team that's actually housed in its own C Corp company. And this does a few things. One, it creates an incredible level of focus at that asset. So you have scientists that only work on that one target. That's really important because in genetic diseases, it's often a very idiosyncratic target biology or set of assays that you need to run your program. So we thought that was really important. And the second thing it lets us do is be nimble and agile. So we don't have layers of bureaucracy on decisions that we view as unimportant to the overall success of the program. We let the project teams actually call the shots on the key decisions because they're best positions to know what's right anyway. So that's actually resulted in a great efficiency in our pipeline. I think we've done in the last 7 years about 15 INDs or 15 molecules into the clinic. We usually do these in about $10 million or less, which is, I haven't heard of any company that can get close to that. And in the clinic, where it's really hard to be efficient sometimes, I actually think we're doing a pretty good job there where we can get from kind of idea to clinical proof of concept. Now, this depends sometimes on the disease state, but we've done this in between $50 and $80 million in most cases. So that's idea to I've got a proof of concept read in my drug and I know it works, so we've got a green light to phase 3. So where are we now? We have got 2 products that we've had approved, smaller diseases, not huge sales opportunities for us, but we've licensed the commercial rights to parties that we think can extract a lot of value for patients. And we're by the end of this year, I think we'll have 4 phase 3s ongoing, a couple more potentially moving to phase 3 behind that. So we're kind of on the doorstep of a lot of revenue if we're successful here. If even half our programs are in phase 3 are successful, we could really be accelerating a lot in terms of our growth trajectory here. So happy to get into discussion with the panel.
Great, thank you. So I'm happy to open it up now if there are questions, but if not, I can also ask the panelists any number of questions that I have about all these interesting business models. But let me turn it over to the audience first.
Hi, everyone. Thank you for the discussion. I'm Mario Romani from Columbia's Tech Transfer Office, and this is a question I think I've been dying to ask all the panelists, but we've talked a lot about mismatched incentives, and so I'm curious from your guys' perspective where you think along the drug development pathway we should invest funds once they become available. Is it focused on indications in rare cancer for existing therapies where maybe there's not a lot of interest from the investment side? Or should we focus on early stage research and new compound development?
Marshall?
Coming out of the university background and 40 years of that, and you're doing tech transfer at Columbia, at Vanderbilt, what we did is we focused on getting things ready to license. Doing spin-off companies from universities, those don't always go particularly well because just because you're good in the lab doesn't mean you're going to be good at running a business. So what we focused on, and then we've also done the same thing at Children's, is trying to get things, what I would call shelf ready for that. And that seemed to pay off pretty well. I'd be curious to see what others think.
I can start. I mean, I think from my perspective, the important thing is trying to have something that you could hand off to a company that's either got reduced risk on the target or some kind of path to it once we're willing to bring it into our kind of labs. And, it's not always the same. So, I think the issue is we're willing to start early with a high throughput screen working with an academic. That's expensive. You might not get hits from a screen. Usually you do, but they might not validate, right? So that's extremely risky. But if we know the target's good, we know there's an unmet need, we'll invest in it. Well, we've done this before. On the other hand, sometimes there's a target that is less validated, but we know that if it happens to work, there's a huge payoff at the end. For example, a disease that may be genetic, but it's very common, and there's a huge amount of patients we could help, therefore a huge financial opportunity for us. So I'm sorry, I didn't answer your question directly, but hopefully that gives you a flavor of how we think about it.
And maybe I'll just add a perspective here too. So from American Cancer Society, over the course of our history, we funded over $5 billion in research in cancer, 25,000 grantees, 1,100 different institutions. One of the things that we're doing at BrightEdge is looking at those data that have been generated by our grantees and those grantees that are generating data that could have commercial relevance. We haven't gotten to the point where we're seeking to extract those and do company creation activities, although we're starting to with some partners. But really understanding sort of how much of that research is going towards new target identification or sort of repurposing and other opportunities. We're definitely seeing everything sort of across the board. In prior roles, I would argue that the new target with clear IP opportunities, right, is just the cleanest path to sort of a commercial opportunity. I think that's still true for many of those reasons. But as I think about things now, right, with the sort of impact mindset that we have at BrightEdge, we're really thinking about both the financial return potential for the things that we're investing in, but also the impact to patients. And if something does have a repurposing opportunity, now there's a lot of, sort of stumbling blocks potentially in the way to enable that. But you could think about a faster road to clinical proof of concept. You can think about, broader accessibility of those indications of that innovation. We also have, investments in companies that are looking at sort of I'd say a combination play of not of combining therapies, but looking at the repurposing, but then how do you kind of jumpstart then the new chemical entity development and make small modifications that improve upon the specifics of that mechanism to then kind of move that opportunity and to have more focus on that important mechanism that could be repurposed for another indication, but also create some new IP along the way so that it does have an attractive sort of business model attached to it for whoever the next partner may be for that. So it's a great question. There's not a singular answer for it, unfortunately.
I mean, we used to bootstrap a lot of things. You'd find a rare disease indication for something. You'd use that to get the process rolling, knowing that that same compound or whatever might actually have utility in a broader population, exactly what you're talking about there. And that can be actually very attractive to get things rolling.
Chris?
Tom, yeah, that was really interesting idea of having a basket of rare diseases to sort of de-risk the whole enterprise. So two-part question. The first one's pretty simple. How many diseases do you have to have in your basket to sort of de-risk to the point where you can be fairly certain you're going to be successful in one? That's the first question. And the second, I think you said that you set up each group as a separate entity. That's interesting because I imagine that most groups are going to be doing pretty much the same thing. They're going to be looking for a good model of their disease, and then they're going to be trying to repurpose approved or de-risked drugs. So there's going to be a huge amount of overlap in what they're trying to do. Do you have a way that they all share screening facilities and libraries, et cetera, et cetera?
Yeah, so on the first one, I can't get this wrong because it comes from one of your papers. I think it was about 20, if you focus on genetic diseases, is enough to get you the diversification you need. So that's why focusing on genetic diseases was a great way to try to apply these principles, into practice, because 20 programs, it's a lot, as I've heard about a million times from equity investors, and it's too much for some people, but it's doable under one roof. The amount of capital you need every year is raisable, it's financeable. So that's the first question. The second question is an interesting one. So yeah, one of the ways we actually, so let me give you a little bit of the backstory on the actual reason we set every program up in its own entity. That was rooted in, originally we didn't know that BridgeBio was going to be the public entity that was financing, getting the capital and deploying it. Originally, we had the idea that the subsidiaries might go out and raise independently, and we still very much have the flexibility to do that. We found that to be inefficient, though, because it actually takes a ton of management time, cash, and just overall bandwidth to go raise money, to have 20 different teams raising money. It's crazy. So we have a small group of people that do that. We just deploy it. In terms of the resourcing question you asked, yeah, that's a really important way in which we gain a cost efficiency. So we tend to think about it as, and this has also evolved over the years. How do we think about what's shared central versus what's sitting at each program? And the general rule now, and this has been pretty stable for the last couple of years, is if it's something related to the specifics of any program, the target biology, whatever, you hire that specifically in your team. So you want to hire a biologist that knows, FGFR3 if you're working on achondroplasia. You want them only working on that because they know the literature, they know the assays. Sometimes we'll hire them from the big labs in the space, or even give the lab money to have a postdoc that can run assays if they've got them in their lab. So there's that kind of thing. If it's something that's more generalizable, for example, medicinal chemistry is a great one. I think we've got some of the best chemists in the world, but a chemist doesn't need to know all of the background on a target. They just need to know, what's my assay that I'm screening against, and what are the properties of the structure and all that, and they can kind of do anything. And so we spread chemists across programs, and then of course we have the huge benefit of having one centralized G&A infrastructure that we spread across all the programs, and that's variable as well. So as we scale up and down, that can shift in size so that we're never hurt by overextension of fixed costs on lab space or anything like that.
Let me actually elaborate on that, because there's an interesting connection to some other people in the room. So the reference to 20 companies that Tom mentioned came out of a paper that I wrote focused on rare disease therapeutics. But my interest in it was not because of the patient perspective. At the time, it was because rare diseases has a really unusual financial characteristic. And that financial characteristic is, because rare diseases are so different, and because the therapeutic interventions are often so different, the success or failure of one rare disease program generally has nothing to do with the success or failure of another rare disease program. This uncorrelated aspect has a very interesting property, because it turns out that it allows you to reduce the risk if you have enough of these shots on goal, to use a hockey or a soccer term. And to make this plain, I give my MBA students an exercise every year. I ask them, can you name me 5 financial investments that are each mutually uncorrelated with each other? You can name any 5 you want: stocks, bonds, currencies, commodities, real estate, private equity, whatever you want. But 5 financial investments that are pairwise uncorrelated. I challenge you to do that. You will not be able to. Everything in the financial world is correlated with each other, and largely with the S&P 500. But I can name you 20 rare disease assets that have nothing to do with each other. And what that means is you can reduce the risk far faster with a rare disease portfolio than a financial portfolio. So I ran a simulation. It looked pretty interesting, but it wasn't on real data. And somebody suggested that I contact this organization at NIH focused on rare diseases called NCATS. And so I contacted NCATS, and at some point after I reached a certain level of handlers and they did a due diligence on what I was asking, I was introduced to Chris Austin, who was head of NCATS at the time, and asked whether or not he'd be willing to collaborate and allow us to use the NCATS data to run the numbers. And we did, and the numbers looked even better. And a former PhD student of mine read the preprint of the version of the paper, came to one of our CancerX conferences and asked me to redo the simulations, not with my assumptions, but with his assumptions. And after about 6 months, this former student came into my office and said, I just want to let you know, I quit my job yesterday. I'm going to do this. I'm going to build this company. And I got to tell you, that freaked the hell out of me. I've never had that happen. So that was the beginning of BridgeBio. I felt guilty enough that I invested a small amount of money in the friends and family round. As of last week, that investment returned 37x, 3,700% return. So don't anybody think that rare diseases is not a profitable investment. It can be a very profitable investment. And you know what? The folks at BridgeBio, they couldn't care less about that number. What they care about is that they've got 2 approved drugs, another 3 or 4 on the way, and drugs that are saving patient lives that otherwise would not have been developed because they would have gotten the funding to do so.
Well, there's one interesting thing about the financial end of it and how it ties back to patient impact. We think about sales or potential sales for any product as actually a good proxy for patient impact, because the more patients are out there that you can treat with your drug, the greater the sales are going to be. The bigger your impact, your magnitude of effect, probably the higher you can price it, because payers will not push back if you're curing people. So it all ties together. I don't think trying to earn a lot of sales necessarily is a bad thing, if you do it in a responsible way. I know that's a rabbit hole that we don't need to get into today, but we use sales as a proxy for patient impact, and that's our goal.
Question, yeah.
Yeah, so that's, so first of all, that's all been amazing, and thank you guys all so much for doing this. For the sake of being provocative and to pull back a bit to where we are, I want to push back and ask about the scalability of this, because you guys are talking about genetically linked rare diseases. And basically, Tom, what you described is exactly, if I go to traditional VC and I can draw a straight line from here's the genetics and here's the thing, and I'm going to get funded for that. But there's lots of rare diseases like appendiceal cancer that I don't have that genetic link. So how much is the de-risking important for this to work, to actually scale to things like appendiceal cancer or other cancers where the drivers, the pathology, what the right thing to do is? Because how do you de-risk that piece of it? Because you're never going to make progress unless you actually can do that part.
Can I challenge the first part of that? Because actually so we had no programs when we started this company. We had the idea. And what we did was go around to all of the academics that are in the rare disease space that we see publishing great papers in Nature and Science and at all the conferences. And we just asked them, you have chemical matter even in some cases that show that the target is, targetable, a small molecule. It's not a drug, but it shows that it can be done. And they, in some cases, have never been visited by a VC, have never talked to a VC. But maybe because they're not in Cambridge or they're not in, San Francisco. And it may be because the disease they work on is either actually too small for the VCs to care, or it's perceived as too small for the VCs to care. And an interesting example of the latter is probably our most visible pipeline program at the moment is a molecule called acoramidis for a disease called ATTR cardiomyopathy. And when we started on that program, it was a late, advanced lead, literally sitting on the shelf for years at Stanford. No one wanted it. I think Neil and some of the founding team had tried to start a company with it at Third Rock, but no one wanted to do it because at the time it was viewed as a rare disease, and it was viewed as a market that Alnylam and Ionis were going to dominate, and there was no point. Fast forward to today, it's perceived as a molecule that could be a multi-billion-dollar drug. We got it for almost nothing. We gave the scientists equity in it. They've made a ton of money, so everyone's winning as long as the trial works.
So just a couple comments on this from the ALS perspective, right? So I can't really speak to the cancer space, but in the ALS space right now, it is very much what you described, where if you're trying to develop a therapeutic, there is interest in the for-profit capital space for investing where you have Mendelian genetics that ties directly to it. And everything else is kind of left out in the cold. So we, as a nonprofit, there are kind of three axes that we consider when we're figuring out where to invest our efforts, right? So where do you know the most, right? The ideal would be a therapeutic where you know a lot about it, or a target space where you know a lot about it. So maybe Mendelian genetics, where you have impact on the most people, and that's your second axis. And your third axis is where nobody else is working, right? Where nobody else is competing in the space. Those don't exist, right? That's not where that's a hypothetical, that's ideal. So we do invest some of our efforts in the Mendelian genetic space, but not all of it, right? So we're thinking a lot about the sporadic ALS cases where we know less about it, but where we as a nonprofit can make strides to de-risk assets that might then make them more attractive to the for-profit capital space later on, We did that with a therapeutic targeting CD40 ligand in ALS, and we managed to move that into the for-profit space and into phase 2. And to another earlier point, that drug is being developed for other indications as well, which made it more attractive to the for-profit space. So there are ways to do it, you just kind of have to be creative.
Right. The other point that I'd make is that there's a common misperception, particularly among scientists and medical experts, that the level of precision needed for an investor to write a check is the same level of precision that you need to get a paper published in Nature or Science. I promise you that is not true. And the way you know that is because we still have investors that are writing checks for Alzheimer's drugs. And we don't even understand the basic biology of Alzheimer's, never mind the genetic links. So I think that there's lots of opportunity, but the one thing you do have to get right, you need a narrative. You need a clear narrative that investors can understand. And ALS is a great example of that narrative. They have made a lot of strides on the science, but they've made even more strides on the narrative and being able to get investors to understand that narrative. Marty, and then...
Can I throw something in on this list?
Well, sure.
The other thing to remember too is, I mean, we heard this morning that there are actually some germline mutations that are that. Those can help you find models that you can then apply to the rest of that area. Just those aren't necessary. You know, things don't happen usually in a vacuum. So if you've got some germline models, those may give you some things that are extrapolatable when you throw in environmental influences into the rest of the group.
Yeah, I just want to chime into that. It's my lab who discovered the genetic link to appendix cancer, published in JAMA Oncology last year. So I'd like to respectfully clarify that misconception. There is a genetic link. And I think as I spoke on the GAP study, we're really trying to dig deeper into that and understanding how potential partnerships on the venture capital side may help accelerate that is, I think, a really relevant and timely point.
Thank you. Marty?
I'm Marty Tenenbaum from Cancer Commons, and I'm interested in getting input from the financial people about the problem that I think we're all here for, which is how to make progress in appendiceal cancer. I'm not personally interested in getting a drug financed. I'm not even interested in curing a disease. I'm here trying to help Steve Wallman cure his wife. And we have a room full of experts, and we need a model that I think will be able to do some good for his wife on the time frame that she has. And I haven't heard very much talk about that. I did talk to someone earlier who said the best they can offer is have his wife see the best expert and trust what he has to say. But collectively in this room, we have far more expertise and knowledge than any one person would have. And if we can figure out first what the right structure is to be able to put us all together into a collaborative community, a commons, if you will, the question is, how do we finance that? And I would love to hear the panel weigh in on that.
Well, I'll give you some personal experience. When I started off was in the field of urea cycle disorders, which if any of you have heard of it, I will be very surprised. And the 5-year survival when I started in that field was about 20%. 4 or 5 years after we got it organized, we had the 5-year survival up to about 90%. There's actually a period of time before you have a new therapeutic where just by getting everyone together to compare best practices, you know, listening to the talks this morning, there are a lot of folks doing a lot of different things, but it doesn't sound like everyone's saying, here's where our part of this can lead to best practice. Sometimes just getting folks to get their cards on the table, say what they think their best is. Everyone, we used to lock them in the base. We used to call it the lock everyone in the basement model. Because before you have a lot of sophisticated data, what you have is everyone's experience. You get that first, you kind of come up with what are the best practices. Then that also actually generates your next generation of questions for that. But you're at that stage right now where no one's, at least it didn't feel from listening to it like folks are cross-communicating that much on this ought to work, that ought to work. Hey, if we combine this, maybe that will help. That's probably your best way to help your friend's wife.
How do we finance the community?
Fortunately, that's actually the cheapest part.
Right. I was going to also say, I mean, I do think we heard a few things today that I think can be highlighted. So for example, Jim, I'm on his board, but we're talking about TRAC and the study that they've been doing. They're already, they've seen, what I can't remember the number you said, 10 appendiceal cancers with a variety of different mutations around the country. If you are connected to your lab and those patients could somehow also get tissue screened there, it might cross-correlate with that lab and provide additional data, since those are rare tumor types. So getting more patients to you, as well as when patients are identified, getting more patients on Jim's study, and then also having the opportunity to think about, well, we have a study that we've spent a lot of time building with some fabulous, of the thought leaders in rare cancer. How do we piggyback off of that infrastructure? And then how do some of the pharma companies in this room also interact with that study in a way to drive that forward? It's not limited to appendiceal cancer or one type of cancer. It's tumor agnostic. So there is infrastructure, and the question is where do we go from here in terms of connecting the right people? For example, also Corey Painter had with Count Me In, that was a super fantastic way of using data and big data project out of the Broad to identify mutations in angiosarcoma. And so there are opportunities, and I think connecting the dot on a preclinical with a clinical effort with what pharma is doing and identifying the right patients, getting different arms. I mean, match study, connected with TRAC, connected with Count Me In, connected with what you're doing at Vanderbilt. I mean, these are the seeds for actually creating a collaborative process. Once you identify the targets, it becomes pretty easy for the drug companies to then develop the drugs. Like taking, for example, Loxo, what they did with NTRK. If you look at NTRK, there are certain cancer types in which NTRK fusions are in 96% of the tumor types. And you go down, and then, in salivary gland tumors, I think it's like 8%. And then you go down to lung cancer, it's 0.1%. Well, if you first identified it in lung cancer, it would not have gone somewhere, but it's made a huge difference for patients. So I think it's bringing those type of efforts together, and I think that collective effort is in this room, and then the money will follow for the funding. Doesn't necessarily mean patients are going to get all those drugs, which is what I also think is really important to discuss today, but that's,
Raghu?
I was going to ask another question, but Marty's question was so interesting. I'm going to follow up with something else.
Can I ask one more thing?
Okay.
Which is, how many people think a new drug is needed as opposed to maybe there's some combinations of drugs that would be...
I don't think you know.
We don't know.
You don't know. I don't think
How do you figure that out?
this seems like one of the first discussions these groups have had together. I think you probably get a group smaller than 10 together who really are doing a lot of this and have them say, what do we know now?
Can I ask a follow-up question? This is for Dr. Carter. We've talked a lot about the, James, on this side. We've talked a lot about the financing on the small molecule side, but I wanted to get your thoughts on what about the commercialization of the kind of precision oncology support idea. I think, the NTRK example is great, but with the fusion, you have one dominant oncogene and a single inhibitor gives you a great response. And, there's, one way that appendiceal cancer is similar to colon cancer, it's probably that there's redundancy in the oncogenic signaling, and just one molecule is probably not going to work. You're probably going to need combinations, and that the genetics are not going to be single driver. It's going to be combinations working together. We see that with the GNAS and KRAS co-mutations. It's going to be a complex network. And so to unravel that, we're going to need, either whether machine learning or other, kind of complex decision support, that has been done on an N of 1 basis where, a scientist will get involved and do it, but to really fund, the right people to have the knowledge to do that, would need to be profitable. And so can you speak to how that side would be commercialized to really bring the investment?
Well, I don't know that I have the exact answer to that, but I still think in a rare tumor type, I agree with you. Obviously, appendiceal cancer, many cancers they don't have a single driver mutation or fusion that's leading to the cancer. I would say that many rare diseases don't have that either. And I think if we went around the room, everybody could raise their hand with knowing someone who has some type of serious disease where they have no idea how to treat it. So, so I agree with you on that. But I think, this particular disease type is small enough, and there are enough people in the room who are studying it that if you can actually bring, the tumor types, like the patients are incredibly engaged. We've talked about that today, where if you have a rare disease, a rare cancer, something that's urgent, and you don't know how to treat it, patients are incredibly engaged, as are their families. And if you create a network where you say, these are the 5 best research tests, and these are the 5 best FDA approved or 510K approved or LDT available tests that are on the market, and we're going to have this consortium of testing that's done to try to identify. Many of these are AI machine learning driven. You just need enough data in to be able to then identify additional targets. You can't do it on 1 or 2. And so
which brings me back to the main question that I wanted to ask. GPT chat and machine learning and deep learning. Why can't somebody in this room actually start using those things to
So
achieve more knowledge?
I'm actually meeting with a group from Microsoft on working on one of the things we have. How do you write clinical treatment protocols for 10,000 diseases when you're discovering 6 new ones a week? And we've actually been playing with that some. It can go down a rabbit hole pretty quickly, and you still have to have expert input on this. It's a nice way to aggregate stuff.
But that's one aspect of it.
Yeah.
I see this as a systems problem where you have information leakage at multiple areas. One is it's hard for somebody to come up with a proper definition of the disease. GPT chat or deep learning can be used there. It could be used for writing clinical protocols, as you said. It could be used for identifying repurposable drugs. So it seems like you need more than just looking at one thing, so you don't go down that rabbit hole.
The question is how much of that data is actually in an accessible format for ChatGPT? You have to realize those things are only as good as what they're trained on. And that's, I think it's not magic. I mean, it's garbage in, garbage out, or if there's no data in there. And so what's sitting in everyone's, hard drives in their computers is not accessible to those things. So please.
Yeah, I just want to add maybe one additional thought here too. So I think the opportunities for AI and machine learning, especially across cancers and thinking about mechanisms that are shared across different indications, there's definitely a potential there. I am not an AI expert by any means. I barely can hold my own talking to ChatGPT. But I think, and like I said, we are definitely seeing some really interesting opportunities there. To Marshall's point, much of the data, though, is not publicly accessible, right? So the last time I was in this room, Dr. Weinstock called me out, so I'm going to call him out. Now, Big Pharma, I think, has an interesting role to play here, and I know there are multiple companies doing some really innovative, collaborative work here, because some of the best data that exists on how different mechanisms may be at play is based on the controlled data that exists from clinical trials. So one opportunity here may be, right, for some innovative business modeling around access to those clinical trial data in a way that can enable repurposing opportunities or repositioning. And my quick definition difference there is repurposing approved drug, new indication, repositioning shelf drug, new indication, better indication. So a thought, I'm going to plant it, and only because he's sitting right there and called me out the last time. David, I don't know if Merck is doing anything specifically in this regard that is informative.
I think we, like everyone else, are looking for all the opportunities we can with our own drugs. I do think there are models, and I spoke to somebody during the break, for ways in which non-competitive data could be shared in even a financially potentially profitable way, where if that data got licensed out or if there were discoveries from that data, there would be opportunities. There are, going back to something that was said earlier, I will say there's a problem with human psychology where we're all more afraid of losing something than we are of not gaining the same amount. So there's a real protective mechanism that kicks in. Mm-hmm. I wanted to say one other thing, and it's more of a comment than a question, but I think it goes back to what Matthew was talking about. there's a little bit of conflation here between numerators and denominators, right? So the denominator is all these rare diseases. The numerator is the ones where we develop some kind of advance. And so you could have 20 or 50 or 100, and you might make no advance in appendiceal cancer. You may not even go after appendiceal cancer because you don't consider it a low-hanging fruit. And then the ultimate numerator is somebody's friend with appendiceal cancer, and they want to know, what are you going to do for us right now? And most of the answers that we have to that are, and then most of the answers that we have to how are we going to ultimately improve the situation are maybe in 2027, 2029, 2031, we're going to have some advances if we can make changes now. So I do think we have to be really careful about exactly what we're trying to do here.
Right. So I have a response to that, but rather than go into that right now, let me give more questions, and then we'll continue it. Larry?
Yeah,
Larry?
thanks. If we had another hour and a half, I'd love to actually talk about innovative clinical trial design,
Yes.
which I know that Jennifer is very interested in, and we've talked about it a lot. What I see with people with difficult to treat cancers, rare cancers where we don't have good therapy, is they end up going to alternative medicine practitioners or other quacks. And that's a tragedy, I think, for them, but it's also a tragedy for the field because we're not learning from those patients by doing things. So I just want to mention that clinical trial design is a very important part of this thing. And for those of you who are not clinical investigators, eligibility requirements, even for first-in-human trials like phase 1, phase 2 trials, often exclude exactly the kinds of patients that you really want to study, the people who are very sick. And that right now, with the current system, we're losing those individuals and not learning from them. But the question I want to ask is that, and I want to get back to appendix cancers and a lot of these other rare diseases, the problem in many cases is late diagnosis, which used to be the problem in ovarian cancer, still is a problem in ovarian cancer to a lesser extent now, but it used to be, if I go back into the 70s, 80s, a much bigger problem with breast cancer. So the question is, are all of these innovative methods of actually funding, advances, are they applicable to diagnostics? That's my question.
Well, I would say funding for molecular diagnostics is much less than funding for new drugs, definitely. Getting molecular diagnostics to patients is a real challenge in terms of reimbursement. There are many companies now that are looking at early detection. And they're looking at it by circulating DNA. We're back to looking at some CTCs again, which is a little back to the future. There's RNA approaches, there's exosome approaches, there's all sorts of approaches to looking for early detection. It would be fantastic if one of those really works. I think a lot of them are honed in right now on early detection of lung cancer. As you said, Larry, I would be super excited about some of the other ones like ovarian and pancreatic, but this suffers some of the same financing problems, which is that those cancer types are rare, and so then are rarer. They're not really rare, but then to sell enough of those tests to be profitable or to make enough money, there's less of an opportunity.
Payoff. The modeling that you did is that if you take a lot of shots on goals, if you score a goal, you score big enough to be able to compensate for what you lost. With diagnostics, where the profit is much lower, I don't know whether the model would really hold, and yet one of our biggest problems in rare cancers and all cancers is diagnosis.
Yeah. Diagnostics might have lower profitability, but they also have lower risk. So again, it's the Sharpe ratio. And I do believe that there's a business model where you can have diagnostics, a portfolio of diagnostics that could be very valuable, but you need to have a theme and you need to inspire investors to believe in that theme. And there are examples of that, liquid biopsy companies and cell-free DNA companies. So I think that that is something that's worth exploring.
I'm going to throw in a word of caution here from the field of biochemical genetics, where we've been doing newborn screening since the 1960s for very small population diseases. And there's been a big push in expanding newborn screening for these rare biochemicals, lysosomal disorders, and you know exactly what I'm talking about here, is the ratio of patients that you may detect, but also your false positive rate can actually swamp your system. So it may be that, yes, you might pick up more patients with the condition, but you are going to have probably an order of magnitude number of patients. You're going to suddenly say, oh, by the way, you may have rare appendiceal cancer, or take some other form from that. And there's actually a healthcare system penalty for doing that, because it takes a while to sort of, first off, figure out if that patient has it or not, and second off, then get them off the ledge from that. And that's not actually a trivial issue. So one thing we find is whenever we get a new screening tool for newborn screening, we look at that thing really carefully from the standpoint of how many patients are we going to actually tell that they may have something who don't actually, because that actually can be a huge factor. Just keep that in mind.
Right, that's a huge issue for the multi-cancer early detection test.
Okay, yes.
Yeah, I just got the mic. Hi, Dylan Morris. I'm a managing director at Insight Partners. I invest as a biotech venture capitalist in liquid biopsy and oncology and ALS, actually. And I think, in the diagnostic, cost-benefit analysis we're talking about, especially the point you made, Marshall, about false positives. I mean, that's certainly true from a health economics payer perspective, but it's not true from the individual perspective. Because as an individual, I'd way rather run down a false positive than get a false negative or not get a test at all. And I want to make another, I know you're shaking your head, but we have a technology, high resolution MRI, that can catch very small solid tumors that is sold to consumers, in fact, by startups on the coasts, and which is not accessible because of the cost of the healthcare system. And thinking about the impact of that kind of preventative screen on an individual, to me at least, seems worth thinking about financing mechanisms for that. And then when it comes down to specific diseases where the research may not have definitive answers on mechanism of action and the risk is too great for someone like me to invest, I wonder if we should consider something like the focused research organizations that the Schmidt Futures Group has started to fund, where you can maybe, drive through the misaligned incentives, whether it's the lawyers at the institutions defending the institutions or the corporate pharma protecting their data, and go towards a goal of actually addressing need for patients through not necessarily profitable endeavors, but philanthropic. I don't know what you want to say to that.
I mean, I'll just comment on the testing one. While for a patient who is a true positive, that's a real plus. But remember, with a lot of these testing, what's usually the ratio? 4-to-1, 5-to-1? I don't know with the cancers as much. I know with the biochems. So for every patient you have a true, you're going to tell 4 folks, oh, by the way, we think you might have something. And even when you disprove it and say, oh, you don't actually have it, you always end up leaving them with but if anything funny happens, call us back. And now you've created a patient who's going to be hypervigilant for the rest of their life. And that actually does have an impact.
But it's not good if you do a test for Down syndrome and then the follow-on to that false positive is an amniocentesis, and then the patient aborts from the amniocentesis. So there are real medical consequences to.
In cancer, it's even more.
Yeah.
Because you use biopsies and needles.
Yeah. The other thing is, just to be more of a Debbie Downer. So the data for lung cancer screening in former smokers is very clear, about a 20% improvement in overall survival. Those people are cured.
Right.
And then we just published a study, just to sell some more Merck, Keynote 671, that showed a further 20% improvement if you give perioperative Keytruda. So that's 40% improvement in overall survival. That is much, much more than mammogram, PSA, and the national usage among people who should be getting lung cancer screening is 6%.
Yeah.
Right. Yeah.
Yeah, this is such an important point on the early detection, and it's that patient journey of what is the patient actually need to endure to get that screen to go to the next level, and what that drop off rate is between each stage of that, right? And there's so many things that we kind of take for granted sitting in Cambridge, right, and Boston, with the access that we have to these medical institutions, and be able to do that in our own backyard. When we think about opportunities in the early detection, multi-cancer early detection space, no doubt the opportunity and the need there is large, because it is the early detection is still the number one best therapeutic measure, right, because of so many more opportunities that's afforded. But really understanding that how that patient is then going to receive that test result, what they have to do next, and then is that something that is accessible to those who need it most?
Infrastructure
is a big problem, and don't have the exact answer for it, but as we think about things from our perspective, it's certainly one that we pay a lot of attention to, and kind of utilize some of the connectivities that we have to, healthcare systems across the country, both, small community hospitals, but also large NCI designated cancer centers to help address that.
Right, and that's why a lot of the early detection tests are focused on lung cancer, because you actually can make a big impact. But the single cancer early detection test, then you can really maximize for sensitivity specificity ratio to get to really get the right outcomes for the patients.
But just to underscore, when you say big impact, that means different things to different people.
Absolutely.
For David, big impact is like $1 billion or $2 billion a year in revenues. $100 million revenue a year drug is a failure at Merck. That is a very nice rate of return for BridgeBio investors.
Correct.
So we have to keep that in mind. Yeah, Miggy?
Yeah, I just want to bring back to, we're talking about, innovative way of financing. I just want to mention that not some of us, and I think a lot of us felt like we don't have the financing resources. But I felt I would like to ask the group participants, because there's so many smart people here, I was being very impressed, is each one actually brings a non-financial resources to this table to get this the problem solved. So for example, our biosciences, we talked about we can actually use our vascularized model to actually test all the FDA-approved drugs on targeted cancers. So I would just say when at the end of the meeting, I would love to see, everyone, even though you don't have financial resources, but you think about, you do have some valuable commodities to really make this happen. And I would say put our biosciences, for example, on the table, say we do bring a human relevant vascularized model to really be part of that activity. And I think everyone has a play to bring to the table.
Yeah, well, definitely. That's a great question. We're going to get to that. If not at the end of this session, then certainly at the wrap up. So that's a great point. But Karen?
Yeah, hi. Corey Painter. I'm at the Broad Institute and ran the Count Me In program. And I wanted to kind of go back to something that Jen had highlighted after Marty had asked, like, what's the model? What are we going to do? And it seems to me that maybe step one is that in an effort to self-organize, we all come together and just put down on a piece of paper what already exists. Like, I'm on the outside of this field. I have no idea what buckets of real-world data are there, what's available in the space of genomics, what might not be available but we know exists. And if our friends, at Big Pharma had a negative trial, but we could use the data for a synthetic control arm and it's no skin off their back, maybe that gets brought over. But I think until we know what is actually there, what's available, we can't really move forward. We're going to go down a billion rabbit holes. And so, I've heard a lot of interesting things. Vanderbilt, the work you're doing is fascinating. It'd be great to hear more details, like what else could be added to that and how does it get leveraged by everybody in this room? And if there are things missing, like if you wanted to add something in the epigenome, who here is doing that, and can we bring that on board and how does it get funded? But I think once we know what the problem and the scope is, then we can talk about innovative funding models.
Yeah, y'all should be very disappointed if there's not a follow-up to this meeting of a small core group that does put cards on the table and then tries to figure out what do we do best practice now and then best practice moving forward. One lesson we've learned in what I would call the pediatric rare disease field is we get very used to working in ignorance, which I think other medical fields are not as comfortable with. Very rarely is there a publication trail on a disease that we work with. We kind of have to figure out what it is we're going to do and then know that we're going to be building the airplane while we're flying it. That model, I think, applies here to a certain extent. And I think the sooner you get those folks talking, all right, what do we know how to do now? What do we think we know how to do? And then there's that vast sea of we don't know. But you can start chipping away at it that way.
In terms of working in ignorance, try being an economist. So.
Yeah.
Dr. Len.
Andrew, thanks so much. Len Lichtenfeld, formerly American Cancer Society, and now from a company called Jasper Health. But that's, unfortunately, I've got to leave to get to an airplane for another trip. But Marshall, I'm going to come back. I'm going to come to two things. I don't want to be too much of a historian. I'm going to try to be as quick as I can because we could have another day or two session and all the things we've talked about. And Jennifer, I'm thinking about you and the reimbursement issues, because I've been involved in physician payment for a long, long time, sat in the committee, know all the issues, and I don't even think the folks in this room understand how complicated, number of hoops, the CPT codes, the application, the CAC things, the Medicare things. We sat here 10 years ago, and I don't know if you remember this, but I actually I gave the first hello, how are you on behalf of the society, and I said, we have the way. I said it was a takeoff. We have the way, but do we have the will? And if you go back to that time, the reason that Andrew brought that group together was because there had been failures to capitalize on targeted therapies. The first hope being Gleevec, and Larry, the Herceptin moment, in 2004, I think it was 3 or 4 in ASCO, and again with what happened this year with breast cancer and TDX. So back then we had the way, but did we have the will to do the financing to make things happen? I dealt with melanoma at NCI from 1972 until I was there for a short time, but that was my disease. I sat in meetings in 2009 at ASCO, and they're using the same darn drugs then, and all of a sudden, along comes vemurafenib, along comes immunotherapy, and the world changes. So from where we were and having these conversations, the negativity that existed at that moment was substantial. The changes that we've made in the last 10 years have been remarkable, and quite honestly, I have to tell you, I'm thrilled to be here today and thrilled to be part of this group, because it was the optimism that we can find a way through to meet these challenges. Marshall, you said something earlier, and I'll be quiet. I apologize for the time, but I just got to say this before I go. You made a comment about knocking the heads together.
Yeah.
Right? You think back, Myeloma Research Foundation. I don't know if Dr. Flaherty's still here, but on uveal melanoma, I was asked to lead a meeting of the warring factions. And unfortunately, I became ill and I couldn't do it. But that was about 10 years or so ago. And here we're talking about uveal melanoma and the opportunities. We're here talking about a disease and understand the basic is we've got to knock the heads together, bring the people in the room. What do we have? And the key is frequently a patient advocate who says, we will make this happen, sit the people around the table and say, enough. And I think having heard everything that we've heard today, I actually think the money is actually the least of the problem. Yeah. I think the money is the least of the problem. I think it's the egos, the institution issues, bringing it together, make it happen. There's no reason we can't succeed. If we had said we couldn't do it 10 years ago, 11, 12 years ago, we were wrong. And we proved the world wrong. And the successes we're talking about today did not happen without knocking the heads together, without innovative financial approaches. So do not be pessimistic, but it has to move forward with advocacy, commitment, and willingness to come together to make it happen. So I apologize for doing this or taking the time, but I just think it's that important that we learn those lessons and continue to apply them, because we can achieve success if we make our minds up to do it. Thank you.
Right, so, a question here.
Yeah, so my name is Yuan Luo from Northwestern University. I want to propose something that might be a little bit provocative. And so if you think about clinical trial as an optimization problem, and so we do batch update, meaning that we accumulate 10,000 patients, 20,000 patients, and then do the trial, and then implement that knowledge into the guideline. But for rare diseases, we might not be able to accumulate that enough number of patients. So could we do something like an incremental individual update in terms of, let's maybe change the clinical practice guidelines. So while we are treating patients, we're also collecting evidence. And once we have treated one single patient, we might be able to integrate his or her evidence into our current understanding of the disease, and then sort of have this iteration, this feedback loop from every single patient. But that will involve not only the clinical practice community, but also in terms of the pharmaceutical community to maybe change the way how we formalize the clinical practice guideline and also do clinical trials. Would that be something that is worth considering?
That's not novel. I mean, that's how you should do it with the that's how we do it in the rare disease community. Because you start with the best you have, and then you learn going forward. And then, like I said, I use the term building the airplane as you fly it, but it's a continual process improvement. It's almost more engineering than anything else.
But to that point, maybe we can actually spend the last couple of minutes, because I do want to make sure that we cover the one topic that Larry Norton raised, which is clinical trials. All of this is dependent upon the clinical trial infrastructure. And in ALS, they've had a recent success that was driven to a significant degree by the particular clinical trial structure that they used. I don't know, Fernando, if you want to talk a bit about that, or you want me to talk about the Healy trial?
Well, so the success with Amylyx actually happened outside of the Healy trial. But I will speak about the strengths of the Healy trial. So in the ALS space recently, largely led by Merit Sukovich over at Mass General Hospital, she implemented a single master protocol into which multiple groups, multiple sponsors can submit a drug for clinical development. It's a phase 2 program, essentially, and they share a placebo cohort. So it actually gives your patient participants a much higher likelihood that they have an opportunity to be on an experimental drug. And at the end of their duration in the trial, they all get to opt in. There's an open label extension for access. So it leverages really the patient resources. It gives them a lot more opportunity to have potential benefits. And I think it's been, it's that process in place, leveraging a nationwide network. It's really not just at MGH. It's now the NEALS Consortium, which covers, I think it's more than 50 clinics at this point. It really kind of democratizes access as well. And the regulatory agency, I think it's also increased a dialogue between the ALS patient community, clinical community, and the FDA. So it's been kind of a boon for us, really.
The adaptive platform trial was actually pioneered with a collaboration between UCSF, Memorial Sloan Kettering, a number of cancer centers, and the FDA. Janet Woodcock was particularly instrumental in getting this master protocol and the adaptive platform trial designed and implemented at the FDA. And it is a much more efficient way of going through drug screening. You can have multiple drugs in the same trial, and as a patient, this is the kind of trial that you would want to be on, that you would want your mother or sister or a family member to be on, because you get to try a number of drugs until you find one that works. And it's a far cheaper trial to finance, higher probability of success. All the various different boxes are checked. The one drawback is that it's complicated. It's Bayesian decision analysis at the level of that trial design and monitoring, but there are organizations out there that can run those trials.
That was the state of the art in the 2010s. It's not the state of the art today.
Yeah. So maybe one more comment and then we're going to wrap up.
Okay. I mean, adding to that, I mean, there's a lot of things with de-risking this does, because you come in with a regulatory decision. Your design's been pretty much approved when you walk in the door as a sponsor. You have a standing infrastructure. They've already been trained. The budgets are already worked out. I mean, there's a ton of advantage in there. And it's reiterative. They're learning. They're on their lab. So they're iterative process where you learn from the trial. And so, I mean, I'm a strong proponent. I like designing them, so I'm all about that. So. Just getting people to cooperate.
Just straight up, I think that's a great trial design. And Marty, to your comment that was the state of the art in 2010, I think the Bayesian design actually could incorporate molecular profiling. Like the initial battle trial didn't use molecular profiling; it just looked at response. But now that we actually have molecular profiling, the Bayesian design is set up so that you could incorporate that molecular data into the Bayesian framework to have, just like wouldn't make sense to have lung cancer and colon cancer patients, randomized together because we had histology as a divider. But now if we can look at, gene mutation status, KRAS, or a transcriptional feature, like any feature that you're confident on, you could put into that Bayesian randomization and adapt. And so
the big problem, though, is that those trials start with drugs that they're trying to approve in a more efficient way, as opposed to starting with a patient. Because if you think about that the treatment for cancer is not drugs, but combinations of drugs, the combinatorics are infinitely greater. So if you start with a patient, then you have a way to control the combinatorics and learn from that patient to help the next patient.
The problem is you're going to have a hard time financing that.
That was my question. How do you?
So we are now going to get to that point. So first, please join me in thanking the panelists. And if you guys can stay, you can stay because I think you're going to be critical for this next discussion, and you may want to chime in as well. So. So I want to use the remaining time to wrap up. And Steve asked me to do the wrap up, partly because he has an important call that he has to take, but also because I suggested that we say things that he wasn't comfortable saying, but I am. And it's about what we do for and with Steve and Kathy. I echo Marty's sentiment that we like to focus on the patient. And I believe that if we collectively can do something for Kathy, we will be able to do something for many appendiceal cancer patients. So I have absolutely no qualms about focusing on one person. You got to start somewhere. And so what I'd like to start with is a question about how do you get a bunch of busy, talented, competitive people to collaborate? How does that happen? What does that even mean? And in particular, I want to focus on an area that has nothing to do with medicine, because I believe the problem has a very simple answer to it that can be applied to this room, all of you here. And the problem that I'm going to tell you about is a collaboration that involves some financial engineering, although the particular agents involved probably wouldn't describe it this way. It has to do with something called the 2009 DARPA Network Challenge. 2009 was a 40th anniversary for DARPA, the Defense Advanced Research Projects Agency. And as you probably know, many of you have heard of DARPA, they are a particularly effective organization at fostering innovation in a variety of different means. And so they decided that they were going to celebrate their 40th anniversary with a challenge to highlight innovation. And the challenge was this. DARPA announced that they were going to locate 10 large red weather balloons in random locations in the United States, anywhere in the 50 United States. And the challenge was for any individual or group of individuals to be the first to identify the locations of these 10 weather balloons. And the team that was able to email the locations of these 10 balloons first to DARPA would win $40,000 in prize money. That was the DARPA Network Challenge. And so they announced ahead of time they were going to do this. They gave people some time to prepare. And then they started the clock and they said, okay, the balloons are in place, go. And the clock started ticking and various groups started looking for these 10 red weather balloons. It turns out that several hundred teams registered for this contest. And the MIT team won, of course.
Are you in the picture?
I'm not in the picture, okay. But one of my PhD students is. The question that I want to ask you is, how long do you think it took? Now, this is back in 2009. This is before a lot of the social media that we have today. It's certainly before talk but how long do you think it took the winning team to identify the locations of these 10 randomly placed balloons?
1 day.
A day? 24 hours? Yeah. I think. Anybody? An hour. Well, that's 2009. A month. Because these are anywhere. I mean, they could be in a cave in the Rocky Mountains. Who knows? Well, the answer is 8 hours, 52 minutes, and 41 seconds, which may not be impressive today, but back in 2009, this was really impressive. And so the question that I want to put to you is, how did this happen? How did they do this so quickly? And the answer that I give is financial engineering, but they wouldn't describe it this way, the team, Sandy Pentland and the Media Lab students pictured here. Let me tell you what they did. First of all, they did use the internet. They announced online that they were going to enter this contest and they wanted people's help. And they were going to compensate people for their help. And this is the plan that they were going to propose, that they did propose. So what they said was they were going to give away all the money. They're not going to keep any of it. They just want to win. But the way they were going to do that is they were going to dedicate $4,000 per balloon. So that's 10 balloons, $4,000, $40,000. It adds up. So it's credible. So if they win, they're going to pay it out, and they're going to pay $4,000 for every balloon that they discover to the people that helped them discover it. But they had a very specific payment model. The payment model was this. If you knew where the balloon was and you emailed them to let them know, this MIT team, and you were the first to email them, they would give you $2,000 if they won. If you weren't the first to locate the balloon and tell them, but you told a friend about this and the friend emailed them with the location, then they would pay your friend $2,000 and they'd pay you $1,000. If you told a friend who found the balloon and emailed them, they would pay that person $2,000, they'd pay the person in the middle $1,000, and they'd pay you $500. And on and on. And so if you do the infinite sum, you would find that for every balloon, that payment scheme adds up to $4,000, exactly. And so they announced this. And by the time they were done, by the time they had located these 10 balloons, do you know how many people participated? How many people emailed them? It was 10,000 people.
Wow.
In the space of 8 hours, 52 minutes, and 41 seconds, they were able to get 10,000 people to help them. And they paid the money out. Everybody got paid. That's how you get people to help. Everybody gets paid.
And they had to write 10,000 checks.
Not all of them were checks. Some of them, they did not want the money, but they wrote many checks, and that was part of the challenge. Yes, they did. That was the biggest challenge, actually, figuring out how to pay everybody.
And the post.
Yeah. So why am I giving you this example? It's pretty clear that we have the talent in this room, but you all have things to do. You're busy with your own research agendas, your own responsibilities. How do we get you to drop what you're doing and help Steve and Kathy? The answer is pretty simple. You got to get paid. You know, I'm sure that if I asked somebody in this room to volunteer to mow my lawn, I might get 1 or 2 people, maybe my former students, my current students. But if I offered $1 million to the person who would mow my lawn this Saturday, I bet you I'd get a lot more takers. It's a matter of incentives. So the key insight is that not everybody wants to get paid in money. Everybody has his or her own currencies. Some of you want academic publications, tenure, the Nobel Prize. Some of you want no credit at all. You just want to know that you did something good. Some of you want to see patients benefiting. Some of you are politicians and you want to see action from a political perspective and get votes. The point is that if you want everybody to participate, you need to figure out what their currency is and pay them in that currency. And in this case, for appendiceal cancer, I believe that there's more than enough currency to pay everybody. So I agree with Dr. Len that the easiest part of this is to get the money. The hardest part is to organize the talent and figure out what the currencies are and to structure the business model to do that. And so I'm going to end with one proposal, but it's not the only proposal, but just to be concrete about it and how to fix ideas. What we're going to do is to put out we're going to put out a survey that all of you will fill out, an online survey which allows you to express your interest in participating. And let me describe to you what you're signing up for if you are so interested. Those of you who have ideas for how you might be able to help Kathy and, of course, all appendiceal cancer patients, it will be useful for you to submit a simple description of what you think you can contribute. And then what we will do from that perspective is to figure out what ways we can structure a business model where you will be properly compensated for that contribution. For those of you who are at universities or academic medical centers, what it means is that whatever therapeutic you have in mind, you need to talk to your tech transfer officer first. You need to make sure that the IP is locked up and ready to go. Why? You might think that that's an incredibly selfish and mercenary approach. But if you don't do that, then it turns out, and you give away your IP to the world, that's very generous of you, but nobody will invest in that because investors can't make money on that. And therefore, that therapy that you have gifted to the world will never be taken forward, at least not by for-profit entities. So first step, make sure the IP is properly handled, and then a consortium will be created among all of those who say that they want to play, and we will contact all of the tech transfer officers to make sure that they agree that you can play. And then I believe with all of those therapeutics, we can then see whether or not they can be used in sequence or in parallel to help Kathy and other appendiceal cancer patients. And then explore whether it's possible to create an adaptive platform trial to include all of those therapies at the same time for all appendiceal cancer patients. And if you do that, if you have an adaptive platform trial, you have a number of therapeutics that look promising, you have people that know what they're doing in terms of taking it forward, then I believe you can get investors to write checks, either investors that are looking for profit or impact investors that are looking for impact on patient lives, or both. I believe you can do that. Now, it requires somebody to organize all of this. And between Steve and Kathy, myself, and others, I believe that we can put that together. And so those of you who want to participate, if you want to participate by starting your own biotech company and participating by licensing or collaborating with this entity, happy to do that. If you don't want to start a biotech company, you simply want to see your idea helping patients and profiting from that, either in the form of research dollars to your university or royalties, we can arrange that too. There are various currencies that can be constructed to pay for what you're going to do. That's a model. Not the only model, but I know that that model will work. And I know that it will work with appendiceal cancer, given the talent that we have in this room and the talent that all of you can bring beyond this room. I believe that that can work. And so what I'm hoping is that once we get all of your results, all of your survey responses, Steve, Kathy, and the rest of the organizing team will have a meeting, and we will create that smaller group of individuals that Marshall outlined, and we will start planning to figure out how do we get going on this. But I believe that it is possible to do it with all of the talent that you bring to the table, especially given all of the things that we have seen coming out of conferences like this over the past 10 years. We are a lot smarter, a lot more nimble, thanks to all of you and the contributions that you've made. Questions or comments about this before we conclude and hit the bar?
I'd include diagnostics as part of it.
Diagnostics, biomarkers, all of the above. Any way you can help. Don't worry about the financing. Let us worry about that. And obviously, we will be discussing with each and every one of you the particular economic model to see whether or not it makes sense for you. Ron?
did have this adaptive platform trial, which as I'm understanding it, is a consortium of sorts. If you could combine that with the consortium financing.
That's right.
Then you have a built-in non-correlated pool.
Exactly.
Yeah.
That's the idea. No one has ever financed an adaptive platform trial with for-profit investors that actually own parts of the portfolio of the drugs that are in trials. That would be new, but we can do that. We can do that now because we have the business models that have gotten us to this point where it is now feasible.
Andrew, there's a little bit of precedent, but not nearly as good as this, I would say, from the Rare Disease Clinical Research Networks. When we pulled those together back in 2004, we had the patients pulled together, the investigators, and interestingly enough, we were encouraged to find industry partners in that. And did Chris leave already? So, anyway, we actually using those patients pulled together, it was kind of the old Field of Dreams model. You build it, they will come. We took 3 drugs through FDA approval in an 8-year window with under 700 patients total available for a fraction of the normal cost because we had built-in, historic data we were able to use. It was one of the first models where they used historic data for these things. So I think what you're doing should work.
Moreover, if the FDA gets wind of this effort and we have people in this room participating, they will rub their hands and jump for joy and say, how do we help?
Jan will love this.
Yeah, because this is clearly an unmet need, and this is following a page from the FDA playbook of an adaptive platform trial. They wrote that book. They wrote that master protocol. Marty?
So the adaptive platform trial is a great idea, but it's not the first step. The first step and I didn't see it on the slide, which is if you're saying that we're going to start with Kathy and individual things, we have to have the signal-seeking part of this thing, which is the predecessor to the platform trial, so we can find out what hypotheses work, and that's where you bring together all of the diagnostics and workup of all the people in this room who are from that space to try to figure out what's going on in Kathy's cancer, and then we figure out whether we need new drugs or not. My argument is regimens of drugs that may be existing or may be new. But the point being that this is the thing that needs to go step one.
Right.
And then when you have hypotheses that get generated for Kathy, in fact, you'll have more hypotheses than Kathy will be able to deal with.
Right.
So now what you have is an opportunity Kathy gets one treatment, the rest of the hypotheses get, there's a lot of other patients who need something to try, and they get to try those things.
But the idea
of collective learning over the community, and then based on that, we get enough signal that we can decide what needs to go into a platform trial.
And the idea is to do that all at the same time, Marty. I think that Steve and Kathy will be the first to say that they don't want this just to be about them. And I believe that starting with them and including all of these other hypotheses at the same time to be able to build this adaptive platform trial is what we're proposing.
The signal seeking should be on in parallel with
Sure,
all these patients.
absolutely.
And then you get to the,
of course.
It's expensive to put things on a platform.
Of course, it has to be. Yes.
I mean, I think this is a great idea and fantastic. I think coming into this, I was a little skeptical of what would come out of all of this, and I do appreciate this. And I think PanCan has recently run an adaptive design trial as well, which I think is very exciting. And so I would just go back to one of the points, though, and one of the things that we all struggle with patients with peritoneal disease is measurement of response. Mm-hmm. So I think this trial is going to be particularly unique because I do think we have to incorporate biomarkers as well as laparoscopy. So ctDNA, cfDNA, exosomes, all the work that many of us have done. But I think making sure that the FDA actually agrees to that and venture capital, because I think that is going to be perhaps one of our biggest challenges.
Right. So I couldn't agree with you more, but I think this is the kind of challenge that will emerge once you go down the path of designing the trial. In other words, there are, I'm sure, dozens of issues that I've ignored, but those issues will surface very quickly once you start designing the trial, once you start talking to the FDA, And that's how you make progress. There are issues that I'm sure nobody's even thought of that you will now be confronted with once you have the opportunity of an adaptive platform trial. So I think that's the purpose of going through the exercise and then surfacing these issues. But another?
I'll make a comment. Jim, you can actually jump in on this one too. This should make most of you feel kind of uncomfortable. In the world of rare disease, this is sort of our normal operating model. There's a family that gets in our face and says, my kid's sick or I'm sick and you got to go fix this now and you need to know what the hell you're doing. And I don't care if you've got other things to do and other committee meetings. I'm sorry, I'm in front of you right now. And so it is actually a little uncomfortable, but it also is a great motivator because it's the reason we do this stuff at the end of the day. But yeah, so if you are feeling a little uncomfortable, then welcome.
Jim, I think Jim, talk again about track a little bit.
Yeah, I mean, absolutely. And when we all get together in rare disease meetings, and certainly rare cancer meetings, all of our foundations started from a personal story, or most of them did, almost universally. Certainly mine did with my brother-in-law losing his life to a rare cancer. So this is very much in our wheelhouse, to be sure. And I think, what I'm really excited about is I think we have a ready-made structure that can be leveraged here, and that's really why we built the study that we did, so that it could be very, very quickly applied to other disease areas. So this, I think, is a fantastic idea, and really will leverage all the respective skill of everybody in this room.
Well, so on that note, I want to thank all of you for coming, for spending the day with us, and for responding to the survey and letting us know how you'd like to be involved. And I'm going to close by turning it over to Jane, who will say a few words on behalf of Koch, and then the final word goes to Kathy.
Thank you all so much. I don't know about you, I'm just overwhelmed, I'm energized, I'm exhausted. I feel it's like all of the feels today. But what I want to do is give the last word on today to Kathy. Kathy actually reached out to me this morning. She clearly has massive FOMO. She hates missing out on this. But she sent us a message that she wanted to share with us today. And we're going to run that video now. But thank you all for attending. And I really look forward to the future of collaboration on this.
I hope you've had a very creative experience, collaborative experience over the last couple of days. And I just wanted to update you very briefly. Last Wednesday, I had cytoreduction surgery, and the result is optimum, optimum, optimum. Thanks to Dr. Andrew Blakely and his team, Dr. Lou Bryce, many, many others here at National Institutes of Health. My tumor burden is down to 0. All visible disease has been removed. I had HIPEC, I had a colonoscopy, and my ileostomy was reversed. So hats off to Dr. Blakely and his team. I hope that you can go forward here with a good plan on how to collaborate so that we can bring solutions to not just appendiceal cancer, but all kinds of rare diseases. Bye for now. Thank you.